Artificial regeneration is defined as the renewal of the forest crop by sowing, planting, or any other artificial methods. It is reported that nearly about 1.5 million hectares of area of forest are cut every year worldwide. Therefore, in present scenario, the importance of the artificial regeneration is inevitable. It helps to restore the lost ecosystem, reclamation of the land, and improve air quality. Artificial regeneration gives choice to grow tree species as per our need. There are two basic objectives of artificial regeneration. 1. To reforest an area. 2. To afforest an area. Object number 1, reforestation of area. The reforestation of area where forests were removed or destroyed for the purpose of agriculture or urbanization. Objective number 2, afforestation of an area. In this one, establishing forests in an area that have never been forest. Basic steps involved in artificial regeneration process. 1. Choosing of tree species. Before going for plantation of any tree species, the purpose of growing should be known. The tree species are selected according to the human needs such as protection, production, aesthetic value, and other socio-economic considerations. Climate and microclimate. Tree species should be chosen as per the prevailing climatic and microclimatic conditions of that area. Soil requirements. It is important to select the tree species as per prevailing soil conditions. For example, wet soils, salix species, that is populous species, etc. should be taken. 2. Waterlogged soil, Eucalyptus robusta, Eucalyptus saligna, etc. should be taken. If the soil is sandy loam, Albergia procera, Acacia nilotica, Dalbergia sissu to be taken. Marketing facility. Availability of market to sell the logs or the tree product also to be taken into consideration before selection of any tree species for planting. Growth rate. Fast-growing tree species are economically viable. Planting of these tree species give more return. For example, Acacia nilotica, that is bubble tree, Lucina leucophala, that is subabul tree, Melia azederac, that is Malay vembu in Tamil, Melia dubia, that is mahaneem, Grevillea robusta, that is silver oak, Populus deltoides, that is poplar tree, Salix species, that is willow tree, etc. can be planted. Availability of exotics. It means an organism established in an area which is not at all the native of that area but has its origin in some other region. For example, eucalyptus species, Lucina luphala, Robina pseudacacia, Populus deltoides, etc. Site conditions. The site is the complex of physical and biological factors of an area that determine what forest of other vegetation may take place. Succession. The succession is the gradual replacement of one community by another in the development of vegetation towards the climax. Cost of growing. This is also a very important factor affecting the choice of species. Availability of seed or propagation material. The seeds have to be collected from CPCs. Choice of method. The success of artificial regeneration depends to a great extent upon the choice of method. There are mainly two methods of regeneration, namely sowing and planting. 1. Sowing. Sowing is the process of scattering the seeds in a particular place, for example, nursery bed, field, or forest. Advantage of sowing. It is the cheapest method and costs less. Sowing is direct method and no other complications. It takes less time and thus the work is completed soon. In sowing method, there is no question of disturbances of roots. Sometimes sowing is done directly in the forest and hence it does not require any nursery. The sowing is the simplest and less cumbersome. Planting. Planting is another method of artificial regeneration. However, planting is described as the transferring nursery stock to the planting site. Advantages of planting. More success, less seed needed, no damage, cheaper weeding. Disadvantage of planting. Needs nursery, disturbance of roots, time consuming, need of skilled labor, and high costs. Site selection. The selection of site is also a crucial factor in artificial regeneration. There are several factors which affect the site preparation. Following are the factors which are essential to carry out the preparation. Ground cover, that is space available for regeneration. Physical factors such as topography, exposure, soil type, erosion hazards, size of treatment area, and access. Preparation requirement. To create a suitable environment for establishment of desirable species. Manpower and equipment. The site selection and preparation methods require good skill and proper equipment. External constraints such as legal responsibilities, smoke management guidelines, proximity to sensitive areas, and the attitude of adjacent farmers or landowners. Spatial arrangement. This is also called as spacing. Artificial regeneration by vegetative method. Planting materials besides seeds, for example, bare food seedlings, containerized seedlings, cuttings, layerings, rhizomes, suckers, offsets, bulbs, corms are also used as vegetative propagation materials. Propagation by cuttings. Cuttings are of two types. 1. Stem cutting. 2. Root cutting. First, stem cutting. Very few species respond well for this method. The species which are easy to root are suitable for this method of planting. Particularly, the species of dicotyledons group having active cambium layer, for example, shisham, nimbara, drumstick, mulberry, inga dulcis, dhaman, pangara, pimpal, and banyan, etc. Depending upon the maturity, the stem cuttings are grouped into hardwood cutting, that is, mature woody branches are used, softwood cuttings, where recently mature branches are used, for example, mulberry, inga dulcis. Root cuttings. Roots are used for preparation of cuttings, for example, sandalwood, pangara. Stumps. In a few species, stumps are used for planting, for example, teak, shivani, shisham, cassia species. Stumps are easy to transport, require less space, and can be transported to long distances. These are prepared at the time of planting operation or just before planting operation. Fresh, uprooted seedlings are used to transplant easily. 20% of stem portion and 80% of taproot is kept while preparing the stump. Fine-edged knife or implement is to be used so as to avoid the damage. Stem portion is to cut 5 to 6 cm above the collar region and remaining portion of the roots are cut to prepare stump. The stems are then packed in bundles, keeping stem portion on one side and roots on other side. The stump should be transported immediately. For transportation, stumps are covered with moist gunny bag cloth to avoid desiccation. These stumps are planted on the start of monsoon after 3 to 4 rain showers when soil becomes sufficiently moist and soil temperature are warm. Stumps are planted by preparing small holes in slating portion with the help of crowbar so that new shoot will rise straight. Then the stumps are inserted inside and soil is pressed firmly so as to avoid water stagnation in the hole. The cooler region is kept just near the soil surface. Root suckers. Root suckers can also be used for planting purpose. For example, pomegranate, kokum, anjan, shisham, nimbara, pangara, ethrona, etc. The layers, grafted plants, budded plants can be used to prepare planting material. These are only used in forestry for conservation of superior genotype. It is used for commercial plantation as they are short-lived, spreading and do not develop long straight trunk, particularly suitable for timber purpose. Quality planting stock production techniques. The quality of planting stock is the degree to which that stock realizes the objectives of management to the end of the rotation or achievement of specified sought benefits at minimum cost. Quality is fitness for purpose. Clear expression of objectives is therefore prerequisite to any determination of planting stock quality. Not only does performance have to be determined, but performance has to be rated against the objectives of management. To produce the quality planting stocks, the role of good nursery cannot be ruled out. Containers, nursery media, propagules, water, fertilizers, chemicals, electricity, tools or equipment, skilled labor are major inputs to the nursery. Good infrastructure in nursery, that are mother bed, raised or sunken beds, glass or greenhouse, shade net house, hardening chamber, will put additional facility towards the production of QPS. Techniques for propagation. Sexual propagation. Seed is the product of sexual propagation and it has three major parts. Outer seed coat that provides protection to seed, endosperm that is the food storage of seed, and embryo, young plant itself. The collection of mature seeds from candidature plus tree CPCs to produce healthy, vigorous seedlings is required. Giving pre-treatment to seed before sowing facilitates to break the dormancy to ensure rapid, timely, and uniform germination. Some of the pre-treatment are soaking in cold or warm water, scarification, that is by acid or mechanical method, heat treatment, chemical treatment, alternating with wetting and drying. The pre-treatment time is depending on morphological and physiological characters of the seed, and it varies with species to species. Advantages of seed origin plant. Sexual propagation method is easy and less expensive. Trees become harder, disease resistant, and have longer lifespan. Rootstocks for grafting and budding. Polyembryonic varieties, more than one seedling from one seed, can be propagated by seeds. However, there are disadvantages. It takes a long time to bear fruits. Quality of the plant cannot be improved. Plants propagated sexually are large in size. Thus, the cost of manuring, pruning, and spraying increases. Genetic purity cannot be ensured, and identification of sex is not possible in seeding stage. Asexual plant propagation. In this method, stems, leaves, roots of the plants are used to propagate new ones. Cuttings, air layering, budding, grafting, and micropropagation are the methods to raise the quality planting stocks. Cuttings. Stem cuttings, generally 6 to 8 inches in length, are collected from superior genotypes and after treatment with fungicide and rooting hormone, that is IBA, NNA, etc., put in the nursery bed for rooting. Air layering. This is the method of propagating a plant by girdling or cutting the bark around the branch or stem and packing the area with a moist medium or sphagnum moss after applying some root promoter so that the stem or branch can be removed and grown as an independent plant. Budding. Budding is a form of asexual reproduction in which a new plant develops from an outgrowth or bud due to cell division at one particular site. The new plant remains attached as it grows. Later it can be separated from the parent plant only when it is mature. Grafting. In grafting, two plants are used to develop a new plant with combined traits from the two parent plants. In grafting, the scion is the above ground part of one plant. The scion is attached to the stalk, which is the rooted plant of the second plant. Micropropagation. Micropropagation is the practice of rapidly multiplying stalk plant material to produce a large number of progeny plants using modern plant tissue culture methods. Plant tissue culture is a collection of techniques used to maintain or grow plant cells, tissues, or organs under sterile conditions on a nutrient culture medium of a known composition. Different techniques in plant tissue culture may offer certain advantages over traditional methods of propagation, including the production of exact copies of plants that produce particularly good flowers, fruits, or have other desirable traits. To quickly produce mature plants, the production of multiples of plants in the absence of seeds or necessary pollinators to produce seeds, the regeneration of whole plants from plant cells that have been genetically modified, The production of plants in sterile containers that allows them to be moved with greatly reduced chance of transmitting diseases, pests, and pathogens. The production of plants from seeds that otherwise have very low chances of germinating and growing, that is, orchids and nepenthes, to clear particular plant of viral and other infections and to quickly multiply these plants as clean stock for horticulture and agriculture purpose. Plant tissue culture relies on the fact that many plant cells have the ability to regenerate a whole plant, that is, totipotency. Single cells, plant cells without cell wall, that's protoplast, pieces of leaves, stems, or roots can often be used to regenerate a new plant on culture media given the required nutrient and plant hormones. Meristem culture. In meristem culture, the meristem and a few subtending leaf primordial are placed into a suitable growing media. An elongated rooted platelet is produced after some weeks and is transferred to the soil when it has attained a considerable height. A disease-free plant can be produced by this method. Experimental results also suggest that this technique can be successfully utilized for rapid multiplication of various herbaceous plants. Callus culture. A callus is the mass of undifferentiated parenchymatous cells. When a living plant tissue is placed in an artificial growing medium with other conditions favorable, callus is formed. The growth of callus varies with the homogeneous levels of auxins and cytokinins and can be manipulated by endogenous supply of these growth regulators in the culture medium. The callus growth and its organogenesis or embryogenesis can be grouped into three different stages. Stage 1 is rapid production of callus after placing the explants in culture medium. Stage 2, the callus is transferred to other medium containing growth regulators for the induction of adventitious organs. Stage 3, the new plantlet is then exposed gradually to the environmental conditions. Suspension culture. A cell suspension culture refers to cells and or groups of cells dispersed and growing in an aerated liquid culture medium. Cytokinin induces adventitious buds in kiwi fruit in a suspension culture in about a week. Embryo culture. In embryo culture, the embryo is excised and placed into a culture medium with proper nutrient in aseptic condition. To obtain a quick and optimum growth into plantlets, it is transferred to soil. It is particularly important for the production of interspecific and intergeneric hybrids and to overcome the embryo abortion. Protoplast culture. In protoplast culture, the plant cells can be isolated with the help of wall-degrading enzymes and made to grow in a suitable culture medium in a controlled condition for regeneration of plantlets. Under suitable conditions, the protoplast develops a cell wall followed by an increase in cell division and differentiation and grows into a new plant. The protoplasts are first cultured in liquid medium at 25 to 28 degrees centigrade with a light intensity of 100 to 500 lux or in dark, and after undergoing substantial cell division, they are transferred into solid medium congenial for morphogenesis. Many horticultural crops respond well to protoplast culture. Advantages. Micropropagation has a number of advantages over traditional plant propagation techniques. The main advantage of micropropagation is the production of many plants that are clones of each other. Micropropagation can be used to produce disease-free plants. It can have an extraordinarily high fecundity rate, producing thousands of propagules, while conventional techniques might only produce a fraction of this number. It is the only viable method of regenerating genetically modified cells or cells after protoplast fusion. It is useful in multiplying plants which produce seeds in uneconomical amounts, or when plants are sterile and do not produce viable seeds, or when seeds cannot be stored. Micropropagation often produces more robust plants, leading to accelerated growth compared to similar plants produced by conventional methods, like seeds or cuttings. Some plants with very small seeds, including most orchids, are most reliably grown from seeds in sterile culture. A great number of plants can be produced per square meter, and the propagules can be stored longer and in a smaller area. Disadvantages: micropropagation is not always the perfect means of multiplying plants. Conditions that limit its use include: it is very expensive and can have a labor cost of more than 70%. A monoculture is produced after micropropagation, leading to a lack of overall disease resilience, as all the progeny plants may be vulnerable to the same infections. An infected plant sample can produce infected progeny. This is uncommon, as the stock plants are carefully screened and vetted to prevent culturing plants infected with virus or fungus. Not all the plants can be successfully tissue cultured, often because the proper medium for growth is not known, or the plants produce secondary metabolic chemicals that stunt or kill the explant. Sometimes plants or cultivars do not come true to type after being tissue cultured. This is often dependent on the type of explant material utilized during the initiation phase, or the result of the age of the cell or propagule line. Some plants are very difficult to disinfect of fungal organisms. Clonal forestry technique. Clonal forestry is the planting of vegetatively propagated ramets whose clonal identity is maintained from known and tested orchids for the commercial production of tree. Clonal system as a general system was proposed in 1906 by G. Anderson in Sweden. This technique is to produce quality planting stock, maintenance of genetic uniformity, early flower induction, etc. Vegetative propagation preservation of genotypes get maximum genetic gain when used for regeneration in operational planting program. All the techniques of asexual propagation can be used to establish clonal forests in vast area. Conclusion: artificial regeneration is defined as the renewal of a forest crop by sowing, planting, or any other artificial methods. Nearly about 1.5 million hectares of area of forest are cut every year worldwide. Artificial regeneration helps to restore the lost ecosystem, reclamation of land, and improve air quality. The objectives of artificial regeneration are for restoration of the area and afforestation of the new area. There are many factors to be considered before taking up artificial regeneration, such as climate, soil requirement, marketing facility, growth rate of plants, and cost of regeneration. Artificial regeneration can be done mainly by sowing and planting. The propagation of plants for artificial regeneration may be through sexual reproduction or by asexual reproduction.